So this is what we were learning last, plasma membrane. So these are the important points. It's called selectively permeable membrane. And through the membrane, diffusion takes place, diffusion of water takes place. Then if diffusion of water takes place across membrane, what it will be called as?
Osmosis.
Yes, then it will be called as osmosis. So osmosis means it is a passage of water from a region of higher water concentration to a region of low water concentration through membrane, through selectively permeable membrane. That is called as osmosis. Then we also learned about the three types of solutions: hypotonic, which is dilute solution, which is full of water. In such a water solution, if you're putting one cell, as some of you cell into such a solution, cell will swell up. It will become big in size. Then there is another solution called iso. Isotonic means water that is present in the cell is equal to water which is present in the surrounding medium. So equal amount of water will go out of cell and come inside cell. So cell will not change its shape. It will be same in size. Whereas if you're putting the cell in hypertonic solution means it's a highly concentrated solution. Concentrated means not water concentration, salt or sugar. That will be high concentration. That kind of a solution is called hypertonic solution, where the salt content is more in such a solution. If you put a cell inside such a solution, cell has comparatively more water when compared to the solution. So osmosis will take place from cell to outside. So whatever water is there in the cell will come out. Cell will lose water. Cell will lose water by osmosis. Then what will be the shape of the cell? The cell will shrink.
Shrink.
Yes, yes. Good. The cell will shrink. If you put the cell in hypertonic solution, the cell. Now the same thing we are learning, osmosis within an egg. So to do this activity, first you have to remove the shell of the egg. In the textbook, they have written dilute hydrochloric acid. But at home, if you're doing, you can put the egg inside vinegar, which is having acetic acid. So it will take some time, but you can slowly see that bubbles are coming out, which shows carbon dioxide is coming out because the shell is made up of calcium carbonate. That's why. Such a de-shelled egg, that means when the egg is not having any shell, it will have the plasma membrane outside. Such a de-shelled egg, you have to put it in hypotonic and then hypertonic. When you put it in hypotonic solution, that means which is full of water, then the egg will swell because all the water gets inside the egg. But if you're putting similar de-shelled egg in concentrated salt solution, which is hypertonic, for about 5 to 10 minutes, then you can see egg will reduce in size. It will shrink. Egg will shrink. That's because all the water which is there inside the egg will come out to the salt. That's why egg will shrink. Similar activity can be done with raisins also. Raisins, if you're putting it in hypotonic solution, raisins will become swollen. It will swell. If the raisins are placed in hypertonic solution or concentrated salt solution, raisins will shrink. This is what happens when you place shell egg inside hydrochloric acid or acetic acid. You can see these bubbles. These bubbles are actually carbon dioxide. Then this is a de-shelled egg. You can see de-shelled egg. You can feel the plasma membrane. It will be soft. Now these are the situations when you put the egg in different mediums. This is de-shelled egg. When de-shelled egg is put inside hypotonic solution, egg will become swollen. If the de-shelled egg is put in hypertonic solution, then the egg will become small in size. It will be shrinking. That's why. Raisins, when you put in hypo, raisins will swell. And when you put in hypertonic, raisins will shrink. These were the questions associated with that. Now this was endocytosis in amoeba. Amoeba's plasma membrane can change its shape and it can engulf the food. The word engulf is used for this. We cannot use swallow. That word called swallow. Human beings and animals swallow food, But since it is not the mouth of amoeba, instead of using the word swallow, we use engulf the food. Which process is called endocytosis? Endocytosis takes place in amoeba. Now this is your next activity. In this activity 5.6 in your textbook. Somebody read this.
Mount the peel of a row leaf in water on a slide and examine cells under the eye power of a microscope. Note the small green granules called chloroplast. They contain a green substance called chlorophyll. Put a strong solution of sugar or salt on the mounted leaf on the slide. Wait for a minute and observe under a microscope. What do we see? Now place some leaves in boiling water for a few minutes. This kills the cells. Then mount one leaf on a slide and observe it under a microscope. Put a strong solution of sugar or water on the mounted leaf on the slide. Wait for a minute and observe it again. What do we find? Does plasmolysis occur now?
Okay, good. We'll see how to do. Mount the peel of Rio leaf. Actually, Rio is one red colored leaf. Red colored leaf. It's called as Rio leaf. Pictures I'll show you later. take the Rio leaf and you have to peel it. Peel means take a small upper epidermis part of leaf. Take a peel of Rio leaf. After that, we have to keep it on a slide. This is a glass slide. So Rio leaf actually has chlorophyll pigment. They have chlorophyll pigment, and the chlorophyll pigment is present inside chloroplast. So such kind of a peel of Rio leaf, we have to take it. After taking that, you have to take this peel and keep it onto the glass slide. So now I have kept the peel piece onto slide, and in that peel also chlorophyll is present. Then what they're telling us to add? They're telling us to add on such a peel strong solution of sugar or salt. That means we have to add on that hypertonic solution. So we have to put on that hypertonic solution. That means we can add inside it highly concentrated salt solution. We have to put a highly concentrated salt solution. So what did they see is they saw like this under microscope. When they kept it under microscope, what they saw is since it's a plant cell, cell wall doesn't change its shape. It remains like that itself. Cell wall is same like that itself. So this is the cell wall. No change in cell wall, but there was change inside other things. That is cytoplasm, which is there now. All the cytoplasm started moving away from cell wall. All the cytoplasm and the organelles which is there here, they all started moving away from the cell wall. Even the chloroplast also, since it's a plant cell, chloroplast is present. No? Even the chloroplast also started moving away from cell wall. See, they started moving away from the cell wall, which means that cytoplasm is starting to shrink. Cytoplasm moves away from cell wall, moves away from cell wall. So on top of that, when you put the salt solution, see on top of Rio leaf, salt solution was put, and you know salt solution represents hypertonic solution. So since it is highly hypertonic, what happens is the cytoplasm started to shrink. Cytoplasm started to move away from the cell wall, but cell wall is in same position because it's a plant cell. This kind of property, this phenomenon is called plasmolysis. Plasmo means cytoplasm. Plasmolysis means because it is separating from the cell wall. Cytoplasm is moving away from cell wall when you put the leaf into salt solution. This is called plasmolysis. So here they have written about plasmolysis. Maybe in the description they have written. Okay, so this is what happens when you put the Rio leaf inside highly concentrated salt solution. Next, what we can do is we can also put it in hypotonic solution. So take another peel from this Rio leaf and keep it in hypotonic solution. So another glass slide you have to take, and this time we have to use dilute water. We can use water. We can use hypotonic solution or dilute solution, means which is not having any salt or sugar. In such a solution, when you keep the Rio leaf, what will happen that you have to see. So what will happen is the cell wall will not undergo any change. Cell wall will not undergo any change. Cell wall gives strength to the plant cell actually. But what will undergo change is cytoplasm. The cytoplasm will start putting pressure on the cell wall and it will bulge. It will become swollen, it will bulge and started putting pressure on the cell wall. It will apply force on the cell wall because lots of water got inside, no? That's why. So the cytoplasm will bulge, but nothing will happen to cell wall. So here what happens? Cytoplasm will bulge. No change in cytoplasm will bulge. So everything, all organelles can be seen now. Mitochondria, nucleus, endoplasmic reticulum, everything can be seen in this. And the chloroplast is also there here. Chloroplast will have chlorophyll pigment. Everything is seen here, but you see cell wall, there is no change. Why there is no change is because of this reason. The reason is cytoplasm after taking in water, will put pressure on cell wall. It will put pressure on cell wall and bulge. So when it put the pressure on cell wall, it will become bulged. Bulge means bulge. But what happens to cell wall? Cell wall will put pressure back on cytoplasm. Cell wall will put pressure back on cytoplasm. So since the cell wall will also put pressure, see cell wall is also putting pressure back. Cell wall will put pressure back onto cytoplasm. The cell will not burst. Because of this reason, the cell will not burst. Cell will remain the same. Cell will not burst. Like balloon, it will not burst. So this is what happens in the plant cell. If you place it inside boiling water, boil some water and put the Rio leaves into that. This is boiling water. After boiling the leaves inside water, then you have to repeat the experiment. Then what will happen? That is the question. So first we have to take slide. 2 slides should be there. One slide should have salt solution, that is hypertonic, and one slide should have hypotonic, that means just dilute solution. So like the two slides we have to take, one with salt solution, hypertonic, and the other one with hypotonic solution, and then we have to see what change happens. So what changes happened is it's like this. Actually, no change happened. Why no change happened? Because boiling water kills the cells. Boiling water kills the cells. It is written here, na? Boiling kills the cells. So the plasma membrane will not show osmosis. No osmosis, no osmosis. If cell is killed by boiling in water means they will not show shrinking, swelling, all these things we cannot see. The cell will remain like that itself in the hyper. Only we have to peel. Here you have to peel it, and then you have to do this experiment. This is, you see this and tell me, this is the picture of plasmolysis in Rio leaf. You have to tell me which solution was poured on this.
Hypertonic, ma'am. Hypertonic.
Very good. Hypertonic solution was poured on the Rio leaf. That's why you can see plasmolysis took place. So this is plasmolysis, where this is the cytoplasm. Cytoplasm is red in color because you know Rio leaf is also red in color. No, that's why cytoplasm is red in color. And what you see here, this is the cell wall. This is the cell wall. So the cytoplasm move away from cell wall when you put it in hypertonic solution. That is called as plasmolysis. This is plasmolysis only in steps they have shown. See, slowly cytoplasm is staying away from cell wall. This is more, they are starting to go inside. Last picture, they have completely come away from the cell wall. Next topic is about nucleus. Now every cell, when you were learning about onion peel cell, onion cells, somebody's speaker mic is on, just check your mic and put it off. Okay, when you see the onion peel cells in the microscope, you saw peel cells were like this. What else the cell was having? Like this we saw, here and all it was cell wall. This part was cell wall. So onion peel cells, we have seen structure cell wall. Inside what liquid portion is there? What is the liquid portion called as?
Cytoplasm.
Cytoplasm.
Yes, the liquid portion was called as cytoplasm. And there was a portion which was nicely and deeply stained when you put iodine. When you put iodine, there was one portion which is deeply stained. What is this? What was this? Yes, this was nucleus. So whenever we see the onion peel cells, what is deeply stained? Deeply stained or dark stained. Deeply stained means dark, which looks dark when you are staining it. That is called nucleus. So onion peel cells you saw like this. When you put iodine, according to textbook, they use the solution iodine to stain the cells. Now what they're telling is not only iodine, you can use some other stain also. Like that one stain is called safranin. Safranin is a red color stain. The nucleus will look red in color then when you stain it. Like how I have painted here red color, no? Like that nucleus. Similarly, we can also use blue color stain, which is called methylene blue. So iodine, safranin, methylene blue, we can use to give color to the different structures of cells. Now, when I'm talking about the nucleus, this is a cell. Every cell has got nucleus. Every cell. And nucleus controls activities of cell. That is first point. You can take this down. Nucleus controls activities of cell. Just like for a computer, CPU is there, no, which controls activity of computer. Like that, nucleus in every cell controls activities. Sorry. Nucleus divides when cell divides. So this is amoeba cell. Now amoeba wants to divide means the cytoplasm will divide first. Along with the cytoplasm, nucleus will also divide. And that's how you get two daughter cells for amoeba. So that's the meaning of this point. Nucleus divides. When the cell divides, nucleus also will divide. Then the next thing, what is there is the nucleus is surrounded by membrane. Nucleus is surrounded by membrane. But this membrane is not completely joined like this. There are some pores between this membrane. See, like this, some pores are there for this membrane. So nucleus is surrounded by nuclear membrane, leaving some pores, which is called nuclear pore. So this is the nucleus. This is the nuclear membrane. And some parts are not covered by nuclear membrane. It is called as nuclear pore. Nuclear pore. Then inside the nucleus, there is the liquid portion, which is called as, what is it called as, children? Liquid portion inside the nucleus?
Cytoplasm.
It's called nucleoplasm. Cytoplasm is there outside the nucleus. This area is cytoplasm. Whereas the liquid portion inside the nucleus is called nucleoplasm. Now, inside the nucleus, in the nucleoplasm, there are some thread-like structures. There are some thread-like structures. These thread-like structures are called chromatin thread. It is called chromatin thread. Now, when the cell is about to divide, this is for a young cell. Chromatin thread can be seen in young cell. That means new cell, newly formed cell. When the cell is about to divide, mature, when the cell becomes mature, the nucleus, now I'm going to draw a mature nucleus. This is a mature nucleus. The chromatin thread will become rod shaped like this. When the cell is about to divide, the chromatin thread will become rod shaped. Now here the cell is about to divide, okay? That time, this chromatin thread will be called as chromosomes. It is called as chromosomes. So number of chromosomes in the nucleus differs from one organism to another. Now human beings, we have 46 chromosomes. Like this, so many chromosomes are there. 46 chromosomes are there in every cell, our cell. Now if it is a fruit fly means the fly which comes and sits on banana and all when it is rotting. That fruit fly is having only 4 chromosomes. Like that, number of chromosomes, it varies from one organism to another. Now we'll see what is this chromosome having. It is having something important. The chromosome, I'm going to enlarge and draw this chromosome. The chromosome is made up of a chemical which is called as DNA. It is called as DNA.
Deoxyribonucleic acid.
Ah, very good, very good. DNA looks like this. So every chromosome has got a chemical called DNA, which, as you told, DNA can be expanded and it can be written as deoxyribo. Ribo is actually coming from ribose sugar. One sugar is there called ribose. Deoxyribo nucleic acid. Nucleic acid, that is a chemical which is present in chromosomes. So every chromosome is made up of DNA, and not only DNA. Some proteins are also present. Some proteins are also present along with DNA, because DNA is highly coiled, it can come out fast. So to fix the DNA in place, some proteins are also present on chromosomes. So chromosomes are made up of DNA as well as protein, in which DNA is very important because DNA carries information. DNA carries information for our cell, means what our cell has to do, like it has to grow, it has to make things for us. That information is there in DNA only. So DNA carries information for our cell. It also carries information. It also carries information from our parents. It also carries information from our parents. From our parents. It carries information from our parents. Suppose if I'm drawing here father and I'm drawing here mother. Now this father is having curly hair. Example, okay? Suppose the father is having curly hair. Mother is having straight hair, long straight hair. And we'll draw a child here. This is a child. Okay, now this child, I'm drawing curly hair. That means child has got, you tell me, child has got DNA from father or mother?
Father.
Father.
Yes. Not full genes maybe, not full genes, sorry, not full DNA maybe. DNA controlling his hair came from father. Okay, DNA controlling his hair came from father. But the nose, you see, nose gene came from whom? Father or mother? Father's nose is curved like this. Mother's nose is
Mother.
straight. Yes. Nose DNA came from mother. So DNA means it's a chemical that has information carried from our parents to children. It is carried from our parents to children. Now in between, I use the word genes. People say genes also. Now what is genes is? Yes, genes is a segment of DNA. Any segment of DNA. Now this is one segment of DNA. This is another segment of DNA. This segment of DNA might be controlling eye color. This segment of DNA might be controlling skin color. This segment of DNA might be controlling heart health. This segment might be controlling muscle development, like that. So genes means segment of DNA. Segment means part of DNA. Now in mathematics you might have studied, no? This is called what I've drawn now. What is it called as? What segment is this? What is this called as? In maths we are studying, no?
Line segment.
Very good. Good. Who answered that?
Padmavati, ma'am.
Yes, Padmavati, correct. This is line segment. That means here it is stopped, here it is stopped, isn't it? Like that, if a segment comes in DNA, so many like the segments are there in DNA. Segment of DNA is called genes. And DNA is found on chromosomes. DNA is found on chromosomes along with some proteins. Chromosomes are found inside nucleus. And nucleus is found inside cells. And the importance of DNA is DNA carries information of cell. It also carries information from our parents. So, all right, children, with this, your class ends. You have to read this much topic from NCERT. And check the Google Classroom. Today, homework I will be putting after 1:30 only. Okay, check it and do the homework.
Okay,
Yes,
ma'am.
thank you.
Thank you, Miss.
Thank you, ma'am.